that is nothing but very interesting unit from your syllabus. Unit number 6, seed dormancy and germination. From that we have learned many more things, that is what is actually seed dormancy, then factors which are responsible or imposing dormancy, again the methods used to break the seed dormancy for increasing germination percentage or just to shorten the period of germination. So after that one, again interesting thing we have to learn, that is nothing but the metabolic changes taking place during the seed germination. For your kind information student, I would like to say that what is metabolic changes? Actually catabolic and anabolic changes. The sum of that is nothing but the metabolic changes. So in the sense, catabolism is nothing but the breakdown, for your kind information. Anabolism, that is nothing but the synthesis, and combination of breakdown and synthesis is known as a metabolism. So catabolic and anabolic processes what are taking place during the seed germination, that today we have to learn in this lecture. Actually, major changes are associated with seed germination are mobilization of a stored seed. Keep in mind, most important thing is mobilization of a stored food from reserve tissue to site of embryo. As we know that in the last lecture also I have given you idea just what is embryo. Actually you will have to find out, first of all, open the groundnut seed and white little bit, I will have to say that the white colored structure is there in between two cotyledons of the groundnut. That is the actually embryo, and that is nothing but the miniature of a whole plant. Or we have to say that when we breaks the coconut, at that time white colored small part which we can find in, we have to say that, or at one of the end of that fruit, inside that fruit we will be able to see that is solid structure is there, small white part is there, that is the actually embryo. And that embryo is nothing but the, after germination, that embryo develops into a huge size plant. So that is the actually embryo. So what happens actually during the germination of the seed, the whole stored food material in the cotyledons, or we have to say that in the endosperm, that is supplied to the developing embryo, unless and until there is a formation of a chlorophyll containing leaf, isn't it? Once that is formed, then that actually plant starts its synthesizing food. But before that one, up to the development of that one, emerging of a leaves, whole growth of embryo takes place with the help of a nutrition supplied by the cotyledons or reserve food which is stored in the fruit itself. So that stored food which is in the reserve tissue is mobilized to the site of a embryo, because we know that embryo develops into the plant. So all food material supplied to the developing embryo. Here, that reserve food or stored food is in a complex form, for your kind information, that is converted into a simple form, isn't it? So complex forms of the food material, that is nothing but the protein, starch, carbohydrate, fats, lipids, oblique lipids, in monocots as well as dicots seed we will be able to find out. And that complex food material is converted into a simple form, again resynthesized things, and that are provided to the developing embryo. And that type of changes, isn't it, that type of changes after imbibition of a seed, we will find the changes are going to take place, enzymes are synthesized into that one, isn't it? And that type of changes is nothing but the metabolic changes taking place during the seed germination, means reserve food material supplied to the developing embryo. Now, however, one most important thing, scanty information means there is a less information. Scanty in the sense information is not available. Although that is available, that is not confirmed. So that's a scanty, very less information is available regarding metabolic changes occurring at the endosperm, scutellum, and embryo axis during the seed germinations are there. My friends, actually this sketch I will say that the metabolic changes during the seed germination of a starchy seed monocot maize section is there, and you will be come to know that how the things are going to happen, which type of changes taking place. So here this is a embryonic axis. This total structure is a embryonic axis. Aileron layer is this one, isn't it? Aileron layer is this one. Now, as already I have said to you, whole reserve food material is supplied to the embryonic to the embryo, means embryo is present in embryonic axis. So here that food material comes. Now we will take example, stored food material, that is starch is there. Starch is converted into maltose. Maltose is converted into the glucose, isn't it? And that glucose converted into again sucrose, and that sucrose provides energy to the developing embryonic axis. So like that one, same thing happens in case of, we have to say that the protein also, the protein which is present in aileron protein is there, endosperm protein is there. Protein whatever be stored in endosperm and aileron layer of the seed, that is converted into peptide, that is converted into peptide. Then peptides are again converted into amino acids, and that amino acid develops into amides, and then amides are used for the synthesizing new proteins and enzymes whatever required at the time of developing embryo or germination of a, we have to say that for the growth of, we have to say that the embryo itself, isn't it? So how the changes are going to take place? We know that actually backbone of protein is amino acid. Again that amino acids are resynthesized by the converting protein into amino acid, and again amino acids are incorporated into the enzymes and proteins whatever required for the specific proteins, specific enzymes are required for the embryonic development, that are resynthesized, isn't it? Already we know that the food material is stored in the seeds in the form of a protein, aileron protein, endosperm protein, again in the form of a starch, and that is resynthesized, reutilized. Energy is generated from that one, and that energy, that products new developed are used at the developing embryonic axis. As we have seen in case of starch, as we have seen in case of the proteins, here lipids are there. So from the lipid we know that that is converted into fatty acids, then acetyl CoA, and that acetyl CoA is used for the synthesizing of a sucrose. Sometimes fatty acids again used to synthesize of a triose phosphate. That triose phosphate again used for the synthesis of glucose. From the glucose, isn't it? The glucose which is synthesized by the lipid degradation, isn't it? Into a, that glucose is used in the synthesizing of a sucrose. We know that sucrose provides the energy to the developing embryo, and that comes supplied at the embryonic axis. So such a type of metabolic changes we will be able to find out in case of the monocot maize seed. Once again I will give you idea, my friends, you will have to keep in mind, germination is there. Germination is there in the sense developing embryo at the time after imbibition of a water. That water is used to convert complex food material which is stored in the seeds, in the cotyledons, in the scutellum, in the aileron layers of the seed, that is converted into again such a forms which is used by the embryo at the time of its death. So that are nothing but the metabolic changes taking place during the germination of the seed. Now here we will be able to see metabolic changes during the seed germination of a proteinaceous seed, isn't protein containing seed. What changes are going to taking place? As we know that, my friends, very well. So all the food material, reserve food material supplied to the embryonic axis, isn't it? This is the embryonic axis. Here the protein which is present in a proteinaceous seeds, starch which is reserve food material, lipid is there. Why it is known as the reserve? Because that when that is required at that time, that is utilized by the seeds, that is utilized whenever germination process is going on, that is a reserve food material. For your kind information, when and when that is required, that can be used, isn't it, made available. So here protein converted into amino acid, as already I have said to you, amino acids are the backbone of the proteins. Already proteins are synthesized, but that protein reverse takes place here. Amino acids are that amides, and that amides used for the new protein synthesis and enzymes, the enzymes whatever required for the developing embryo of the seed into a plant, isn't it, for the growth of embryo. Again starch, that is converted into glucose, and glucose is converted into sucrose, and the sucrose, that energy is used by this embryonic axis, because without energy it is very difficult for the growth of the embryo is there, isn't it? Similar case in case of the lipid, you see how the from the lipid acetyl CoA then direct glucose synthesis takes place by using various enzymes. The number whatever given here, 1 2 3 4, these are the enzymes, and that enzymes we have to say that are used here to convert all these things, my friends. That enzymes also most important role playing. Simple form I have given you idea how the things are going to happen, isn't it? Now here also the very much clear idea you will be able to get how the fatty acids convert into lipids. The lipase is there, and that lipase is responsible for synthesis of triglyceride, isn't it? Triglyceride is again converted into free fatty acids. Free fatty acids are oxidized here, oxidation takes place, acetyl CoA synthesis takes place. Again from the acetyl CoA, you'll have to know that converted into a succinate, succinate into maltase, maltase into OOA, PEP, then glucose, sucrose, and germinating seeds, where that is things are provided. My friends, as already I have said to you, for understanding all these things very well, you should know the parts of the seed itself. Dicot, where is the embryonic axis, where is the embryo, isn't it? Where is the endosperm? So you will be able to understand with the help of this the endosperm, isn't it? Major food material is stored into a so endosperm is known as a, we have to say that, food supplying part. So here at the time of developing, so that is a endosperm where the food material is stored, isn't it? So again testa in the sense that is a cover is there. Plumule in the sense we know that whenever embryo develops into a two structures at the time of sprouting of seed, sprouting of a seed, radical comes out, radical develops into a root system, isn't it, which grows in a negatively phototropic and positively geotropic. It goes into the soil and starts establishing and absorbing water and mineral. Plumule grows into stem, plumule grows into a stem, and that whenever that stem develops a leaves, we have to say that, then we have to say that the things are happening, that is nothing but the seed stored material. Whenever food material ends, the synthesizing of a new food material takes place with the help of developing developing leaves containing chlorophyll. Photosynthesis process takes place, what are required food material for the further growth of the, we have to say that, seedling is synthesized then by the plant itself, autotrophic plant itself. Then stored material, food material is not used then. Once the stored material is utilized by that one, the cotyledons becomes very small in size, isn't it? Because stored food material is utilized for the developing growth, energy, all sort of the things are there in case of that one. So friends, this process you have to observe at your places where and where you will be able to see that process at the time of germination. So you have to note down the some observations, you have to note down the things, how the things are going to happen, which parts After germination, imbibition of seed, after germination comes out of the seed, that is a radicle. Then plumule. From radicle, root system develops, and from the plumule, we have to say. So such a things you will have to observe always, my friends. So with this, I think you might have understood about how the seeds itself, uh, after imbibition, at the time of germination, the changes takes place into the seed. That are the metabolic changes are going to take place. As already I have said to you, catabolism takes place, means the breakdown of starch takes place, breakdown of protein takes place, breakdown of lipids takes place. Again, from the proteins, again protein synthesis takes place, that is the anabolism, means breaking and then again synthesize. That's why there is a catabolism, anabolism, metabolic changes we can observe in case of the plant when and when that plant, oh sorry, seed undergoes the germination process, my friends. So with this, today we are going to complete this lecture. Again, I give to you one of the experiment. At your places, you'll have to take some seeds of either of maize, either of groundnut, either of jowar, either of bajra. Soak that seeds for 24 hours into the water, and imbibition, after soaking that seeds, you have to put for the germination. Observe the changes, how the things are going to happen. We know that after imbibition, the volume of seed increases, but when germination will take place of that seeds and develops into a small seedlings, the volume of the seed decreases, because the food material what was stored into that one, that is utilized for the growth of a seedling, isn't it? So that observations will have to make on your own, and if any difficulties there in case of that one, please bring to my notice. And this is the, I think today we have completed our 6th unit 100%. If any difficulties there, go through the books, read books, prepare your own notes, and that will help you to understand again the physiological concepts, because these physiological concepts are there, we can find in case of all organs. So physiology is having a lot of importance in the life of each and every living organism, and you will have to understand the concept. With this, thank you very